Welcome to my world, the world of a medical laboratory scientist. Health is wealth, knowledge is power. Today's topic is worm infection and fever. Ordinarily, when we talk of fever in tropical regions of the world, what comes to mind are those endemic problems that cause fever. Shows with fever symptoms like malaria, and some will even after treating malaria will go on to typhoid because these are sanitation problems. But unbeknown to them, one other very common problem associated with fever is worm infection or worms infestation. You have worms like Ascaris, filarias, Trichuris, Strongylides, Staphylococcus, hookworm. All these worms present with fever, especially when it is heavily loaded in the small intestine or in the colon. And when you now have this kind of worms problems, and you are just treating malaria and typhoid, you find that the fever at times goes and come back again, just because the real cause has not been treated. So my effort today is to try to help us understand the worms that could cause infection, and because their treatments are varied, different based on their differences, I will also highlight what we can do as regards treatment. Children too are very prone because, for example, Ascaris lays egg, over 200,000 eggs a day, and you can imagine it's in the soil. Children put everything, their hands in the floor and what have you, and straight to their mouth, and so ingest these worms, the eggs of the worm, and which eventually move to their small intestine. It could cause diarrhea. It may not last for long. The sickness may present more with fever, and these other complications could confuse whoever is looking at the child. But one thing important is, as we go along into the slides session, I will be able to expose more of the intricacies as regards worm infection and fever. So if you like my presentations, press the like button, subscribe, and more will be exposed to you. The outline, objective, introduction, lab diagnosis, prevention, and treatments are all that I shall be talking about in this video. The objective: to create more awareness of fever associated with worm infection, medical laboratory scientists' effort at differentiating them for efficacious treatment and eradication. The introduction: intestinal parasites are among the most prevalent infections in humans in low and middle income countries. The most important intestinal hermit, both in terms of abundance and disease burden, are soil-transmitted hermits, such as hookworm, Ascaris lumbricoides, and Trichuris trichuria. It is estimated that these hermits infect more than 2 billion people, or more than a third of the world's population. Also, the schistosoma species both are of great public health importance, with more than 250 million people infected worldwide, and an estimated global disease burden of 4.0 million disabilities adjusted life year. Children are generally more prone to heavy worm burdens such as diarrhea, malabsorption, anemia, and fever. So this really shows clearly the prevalence, the morbidity, the need for public health, taking care of all these things, especially because these are more or less things that happen in developing countries. You don't find this challenge more in developed countries because of their level of sanitation and hygiene. But economic backwardness brings about all these things that are preventable. But probably if vaccines, but it's almost impossible right now that vaccines can really take care of all these worms. Yes, the topmost representation is Ascaris worm. It's the largest worm of all these nematodes, and over 200,000 are produced per day by one female, and it moves about, and because of this movement, it finally settles as adult worm in the small intestines, creating that problem, diarrhea and fever. The same thing, the second is the Strongyloides stachylaris, also cause fever apart from diarrhea. It was recently reported as being transfused in the blood, transfused into a patient, causing fever. Yes, this pictorial expression shows the life cycle of hookworm. Hookworm is at the bottom. When it passes out from stool, you can see the larva can easily penetrate the skin. So it pierces through the skin, and you can understand that it sucks blood and creates this dysentery and fever as well. Yeah, this one represents trematode, schistosoma. You can see them in single, and when they copulate, male and female join together. Acute schistosoma is also known as Katayama fever. It's characterized by fever, fatigue of the another symptom that occur after initial infection. This is another one, tapeworm, called cestoid. You can see it just like tape, very long. While generally asymptomatic, severe tapeworm infection can lead to neurological problems, including seizure, and some individuals may experience fever. Yeah, let us look at some research findings. A particular country, Bihar, a cross-sectional population-based survey was performed. Once two samples per participant, and what was found after using all the necessary method, even formal ETA concentration method of stool, it was found that virtually all people, 96%, abhor at least one helmet, and that almost half, 49%, abhor 3 helmets or more. Remarkably, Strongyloides stachylaris infection were widespread with a prevalence of 48%, and Akinola-Anjou, that's hookworm, prevalence was higher than that of Necato, is another species of hookworm. The hookworm species that is often assumed to prevail in East Africa. PCR is a kind of laboratory recent test, gene test studying the DNA, outperform microscopy, which is expected in terms of sensitivity and range of parasites species detected. Now to laboratory diagnosis. These samples are collected. That middle one is the stool. You do the macroscopy, looking at the stool, whether there are live worms there. These are not necessarily things that you need a microscope to examine because they are large enough. The blood can also be examined for antigen study, the antigen antibody test, and at times you find some worms in the urine too, especially the tremadocytosome, hematobium. So the macroscopic examination is the first test looking for adult worms that are viable to the naked eye, and it's advisable that we collect so many samples because at times you may not find the oval of the parasites in the stool, in one single stool. Yes, next is the microscopy. You can see the person examining, looking for the various shapes of egg. And there are other methods of ETA concentration, formula. You can easily use the gene expert, the PCR machine. You can stain the slide, the stool, and other tests. These are all exposed there. Yes, finally, the prevention and treatment. Talking about prevention, we can only mention hygiene and sanitation of the environment because we are in a third world country, a developing country where all these things are very, water is a difficult problem. Even people don't get water easily. Talk less of even using it to maintain, to take their bath, wash their hands regularly so that these organisms, the ova of the parasite, will be washed off and clean the environment. The acronym WASH, Water, Sanitation and Hygiene, remains a vital recommendation if the environment is free from people's feces. Surely all soil hermits, like ascaris and other nematodes, will have their life cycle disconnected from the plethora of worms earlier shown. It is obvious there is no broad-spectrum drug that can be efficacious enough for all. It is therefore recommended that patients with some nematode infection can be treated using mebendazole or pyrantel. Albendazole is the drug of choice for ascariasis, trichuria, trichinosis, cutaneous larva migrant, hookworm, and pinworm infections. Now, the usual treatment for lymphatic filariasis is a drug called diltiazem, which kills both immature and adult parasitic worms. Praziquantel remains the drug of choice for all trematode infections except fascioliasis, for which triclarbendazole is the drug of choice. Ozantel palmite has shown good efficacy in cleaning infections. So all these drugs are very easy, including ivermectin, because there are some of these worms, helminths, that are very difficult to eradicate, especially strongylides, stachyralis, and trichuria. The experience is that you can even have multiple infections, not only one type of worm, the various types. So you can imagine you need the laboratory to help to make sure that yes, you have really eradicated all the worms inside of your intestine or colon. Like I've always said, if you like my presentation, look, there are many videos on this channel. Watch more and subscribe and click the like button. Cheers.